what is 𝗂𝗏𝗴𝗵𝗵𝗁? It's a benchtop-sized screening microscope. From an optical point of view, it's an inverted fluorescence wide field screening microscope. And in combination with software tools, both in-house development, but also we work a lot with open source software, it forms a very comprehensive screening platform that is compatible with a wide range of different samples, ranging from microorganisms. So what you can see here are fluorescently labeled yeast cells. Of course, you can use it for the classic in vitro-based screening using cellular models. But due to its design, both on the hardware and the software level, the system is very well suitable for larger objects. So for the automated imaging and screening of larger objects or larger samples, which mainly means like 3D cell culture applications and small model organisms like zebrafish embryos. So I said it's an inverted automated wide field screening system, and there might be other systems in the market. So what are the main differentiators of the 𝗂𝗏𝗴𝗵𝗵 to other systems? And I think first and foremost is that we have a static sample holder in combination with a mobile optical unit, which means that we never ever move the sample. So we never move the microplate. And instead of moving the microplate in the optical path, we move the entire optics underneath. And the idea was to develop a system that is compatible with motion-sensitive samples. And what is a motion-sensitive sample? At the end, it's anything non-adherent, like non-adherent cell cultures, certain 3D cell culture applications, but also small model organisms that are maybe mounted in a somewhat fragile way in wells of microtutor plates. And by never moving the sample, you eliminate more or less all motion artifacts from your experiments, which is especially powerful and useful when you do time-lapse experiments, but also if you do pre-scan, rescan applications, and I will say a few words about what that means in a subsequent slide. Second important feature is that we have a very precise temperature control in the system based on Peltier elements, so we can heat and cool the system as we can cool. That renders the system also very nice for non-mammalian specimen. But also importantly, the system or the incubation is of low latency. What I mean with that is that we can reach target temperatures within 10 to 15 minutes. So from room temperature to 37 degrees takes about 15 minutes, which also enables you to run like temperature experiments in the device, like temperature curves, looking, for example, at different physiological responses of samples to different environmental conditions. A third important feature is, and I will not cover that extensively today, so I will just mention it here, that we have an open software interface and a scripting interface, which means that in the software of the 𝗂𝗏𝗴𝗵𝗵𝗵, we have a built-in script editor where you can freely program the device using a custom-developed macro language. And also this kind of scripting language can be used to remote control the device from external software applications. Like here you see a screenshot of a 𝗕𝗏𝗏𝗵𝗁 script used to remote control the device, but it's also compatible with any other programming language like Python or Java or whatsoever. And then last but not least, the system can be optionally equipped with a photo manipulation module, which allows you to upscale photo manipulation experiments. So you can upscale it in a 96 well plate format or even 384 well plate format, and run these photo manipulation experiments in a high throughput manner. From an application point of view, I mean, here on this slide, it's just an overview of different applications you can run on the machine. I mean, as a screening system, of course, you can use it for the classic endpoint screening assays, endpoint phenotypic analysis. But as a system is designed to not move your specimen, so we have the static sample holder with moving optics, and the incubation included. It also is so the system is also very nice and very well suitable for upscaling time-lapse experiments to look at more dynamic phenotypes. Like here on the left side, you see a time-lapse experiment of a HeLa cell culture imaged over 48 hours to look at cell division rates. You could look at the formation and development of organoids, look at physiological parameters in small model organisms. Like here you see a blood flow in a zebrafish embryo. You could look at the heart rate in both medaka and zebrafish embryos, for example. or you can look at cell migration, and here in this example, this is immune cell migration again in a zebrafish embryo. So I just want to emphasize, yes, it's a screening system, but I think actually one of the main strengths of the system is to upscale or multiplex time-lapse experiments due to its design. So I said initially the system is designed for larger objects, both on the hardware side, but also on the software side. And on the software side, what we have implemented is a kind of easy-to-use workflow for feedback microscopy, which is mainly used for pre-scan, rescan applications, because if you work with large objects, usually you cannot just go to a random position in a well and image it, because in most cases you would miss or omit the region you actually wanted to image. So you need some kind of means to automatically center on a certain tissue within a large sample, or on maybe a 3D cell culture being randomly positioned within a well plate. And how we realize that on the 𝗕𝗿𝗴𝗵𝗁 is that we typically do a pre-scan experiment at a very low magnification. And here you see an example of such a pre-scan experiment. This is a plate with zebrafish embryos inside and imaged with a 2x objective. And with a 2x objective, you get a very nice match between the field of view and the size of a single well. And then from this overview data, it's very easy to kind of annotate the region you would like to image in a subsequent experiment at, for example, at a higher magnification. And for those of you working with zebrafish who are wondering now why all these zebrafish embryos are oriented the same way, with the head to the left and the dorsal to the top, just to briefly mention, we can also provide these orientation tools with which you can generate cavities in a layer of agarose. And then this agarose cavity has a certain geometry allowing you to position your embryos in a consistent manner, either laterally, dorsally, ventrally, and so on. But coming back to the pre-scan, rescan workflow, so you have your pre-scan data. What you then can do is you can annotate your data. In this case, people were interested in the zebrafish embryonic heart. For those of you not familiar with zebrafish, the heart is located here. And then you can say, okay, that's my region of interest. And then you can, for example, trigger an object detection algorithm. In this case, it's a template matching algorithm, which is a correlation-based search to search for image region in all the pre-scan data that has the highest correlation with this region of interest. You can also define a search region. You don't have to search in the entire image, which will speed up the run and increases robustness. And then what you will get as a feedback are some predictions of the found region of interest. We see here in this example, this worked very nice with 100% efficiency, because although it's a very nice data set with oriented embryos, that's why I selected it for this presentation. So let's say in reality, maybe you may have a few misdetections or the algorithm struggles for whatever reason. What you can also do is you can simply manually correct these annotations, or in practice, you could even click the entire 96 well plate and manually annotate your region of interest, which takes 2 minutes for 96 well plates. So once you have your region of interest, you can instruct the microscope to do something, in this case, center it and image it with a 10x objective. So as I said, researchers here were interested in the embryonic heart, which is here labeled by red fluorophore, and you can see it's always right in the center of the field of view. So I think this experiment nicely illustrates that with this kind of workflow, you can generate very consistent kind of standardized data sets, which are not just aesthetically pleasing, but also facilitate subsequent analysis as it removes variability and randomness from the required data. Of course, it doesn't just work with fish. I mean, here another example for imaging organoids. In this case, we had an EBD chamber, and in each well of this EBD chamber, you have a matrixel dome, which is located at a different position. And in this matrixel dome, you have small organoids growing. And we did a two-step pre-scan, rescan experiment. So first, we identified the location of the matrixel dome, then imaged it with a 4x objective in the fluorescence channel. And then from this 4x data, you can select a couple of regions you would like to image subsequently. And then from these regions, we kicked off a time-lapse experiment imaging organoids over a time course of about 30 hours. That's another example of an organoid imaging application on the 𝗕𝗿𝗴𝗵𝗁. In this case now, it's a scaffold-free application. So you see here like organoids growing in a round bottom plates. And what you actually can see at the beginning of the movie, you just have a cell suspension, which was derived from medaka blastular cell embryos, and then the cell suspension was put into the wells of the round bottom plates and then imaged for 16 hours. And by that, you could visualize the whole process of aggregation, compactions, and then later on acquisition of a certain fate of these cells, which is indicated by the fluorescence reporter gene expression that is coming up. And the reason I like to show this data is not just because it's kind of mesmerizing to watch. It's also, as you can see, that all the samples in this well plate develop more or less at the same pace, and the reporter gene expression is coming up at the same time point in all the different samples, kind of illustrating that we have very stable incubation conditions across the plate in the incubation chamber. And before I hand over to Jürgen to talk about the light sheet technologies, I want to briefly mention our photo manipulation module, which is an optional add-on for the 𝗙𝗶𝗴𝗵𝗁. And so we had a system in place with moving optics, not moving the specimen, and we had workflows for automatically centering objects of interest. So the idea was, can we also use this platform to manipulate objects of interest automatically using light to upscale applications like photo damaging, uncaging, etc. And we did. So we teamed up with 𝗕𝗿𝘂𝗵𝗵𝗵𝗏, who are experts in the field, also a company based here in Germany. And how the photo manipulation module looks like, you can see here in the left lower corner. You see here this white box, which is built into the machine. And in this box, you have two galvos, which are connected to an external galvo controller. You can also connect it to an external UV laser. And what you then can do is you do a screening experiment. You annotate your data with region of interest, and then the system will go automatically and photo manipulate each and every sample. And what you can do with that is illustrated here on the right side. In the left panel, you see these are kidneys of zebrafish embryos. It's a distal tubule of zebrafish embryos. And then when the movie starts, you can see here this little gap. So this kidney was unilaterally ablated or damaged. And then on the device, without going to a different microscope, you can observe the regeneration process in an overnight experiment. And this is currently used to search for modifiers of regeneration in this kind of disease model. Or I showed this image earlier, you can use it to place some burn injury in the fin fold of a zebrafish embryo and then look at neutrophil migration dynamics to the injury site.